Hello everyone, this is Ethan and Ian, and today we're going to be diving into the PDGF pathway.
Picture this. You fall off your bike and scrape your knee. Your body immediately springs into action with a self-reinforcing cycle to form a blood clot and stop the bleeding. Platelets in your blood detect this damage and stick to the injured area. Once attached, they release chemical messages that attract even more platelets to join them. This creates a repeating cycle. More platelets arrive, releasing more signals, attracting more platelets, until enough clot material builds up to seal the wound. Once bleeding stops, the cycle shuts down.
The signaling molecule PDGF is a hydrophilic molecule. Its solubility in water is important to how it works. Because of its hydrophilic nature, it cannot cross the plasma membrane and thus requires a surface receptor. In this case, that is PDGFR. There are several important applications of PDGFR. The first of these, and most obvious, is cell proliferation, but there is also wound healing, as mentioned before. Additionally, it is involved in the production of blood vessels, as well as the general embryonic development process. There are several key advantages to PDGF signaling pathways. The first of these advantages is that hydrophilic signaling is very rapid, and so this signaling pathway takes very little time to go through. Additionally, the signaling pathway fulfills a unique responsibility of cell proliferation, which is a responsibility fulfilled by few pathways within the body. And of course, there are key disadvantages to the PDGF signaling pathway. The first of these major disadvantages is that it's limited to short distances, and additionally, overexpression within the pathway can be detrimental because mutations can lead to tumor growth and diseases like cancer. The PDGF signaling pathway begins with an injury where blood clotting occurs and platelets release the PDGF signaling molecules. One PDGF ligand binds to two PDGFR receptors simultaneously. This is a process called dimerization. Together, the two PDGFR receptors form a dimer. During the autophosphorylation process, each PDGFR receptor provides to another one phosphate group. This phosphate group comes from one ATP molecule where one phosphate is broken off. These phosphate tags serve as flags that other proteins can recognize and trigger reactions that tell the cell to engage in activities like growth, division, or general survival. When it comes to agonists, there are two main agonists, as well as a third artificial agonist that are worth paying attention to. Agonists in general for the PDGF pathway are things that activate or boost the pathway. The first of the two agonists are PDGF-BB and PDGF-AA. BB comes from platelets, macrophages, and smooth muscle cells, while AA comes from fibroblasts and epithelial cells. Becaplirmin is the third agonist, and becaplirmin is man-made. Where there are agonists, there are also antagonists, and the antagonists for the PDGF pathway include two prominent artificial antagonists. The first of these is imatinib, which comes from Novartis and was made during the 1990s. This fits into the ATP binding pocket, thereby blocking or dampening the PDGF pathway. The other type of antagonist is biologics. Biologics are large proteins that consist of living cells. These also block or dampen the pathway. Antagonists have a lot of clinical drug interest.
The pathway can't run forever. Once the wound is completely healed and cells no longer need to grow, PDGF molecules let go of their receptors. The receptors then lose their phosphate groups through dephosphorylation. which turns them off. Finally, the paired receptors separate, marking the complete end of the signaling process. Your body has successfully healed itself and returned to normal.